All right, Megan, can you see my screen and share
Yes.
screen share? Okay, fantastic. Thank you very much. So first of all, I'd like to thank the organizers for the invitation to speak today. Um, my full disclosure here is I have no conflicts of interest, but I'm also not immersed in exposome research. So what I'm going to share with you today is really some of the work that I have been involved with for several months now, looking at virus-virus interactions with a longer-term goal of using these models and tools to actually look at the effect of environmental exposures on immune responses to infection and vaccination. So I wanted to start with my acknowledgements on the front end in case I speak too slowly. This has been an enormous team effort. Prior to March of 2020, I was fully an influenza and vaccine development researcher. As you can see with the footer there, I'm a part of the Centers of Excellence for Influenza Research and Surveillance. But this network was established to be responsive to pandemics. And so when the coronavirus pandemic emerged, many of my colleagues and myself pivoted a lot of our work to address the coronavirus response. And so that included my lab, where we shifted projects to look at developing animal models for coronavirus infection. We were involved in testing novel antiviral drugs and new vaccines, doing preclinical studies, and we have contributed now to vaccines going into clinical trials, which is very exciting and rewarding. And really, it's been an exhausting year for my team, and I'm really excited to show some of this new and unpublished data. I also want to acknowledge collaborators that have helped with this project and our colleagues that have shared reagents, particularly Florian Kramer and Ali El-Beidi, who have developed reagents against coronavirus and shared them very quickly to enable research around the globe. And while I'm not going to be sharing data with them today, Paul Thomas and Jennifer Levy have both are contributing to these projects, and hopefully will provide some answers to questions that I still have even with today's data. So just a quick introduction to SARS as I see it, sufficient for today's talk. I want to answer what it is, where it came from, how does it spread, and what is COVID-19. And so the culprit is actually an interesting virus that I have studied prior to 2020, but not really extensively. It's a negative sense RNA virus, a positive sense RNA virus. Flu is my negative sense friend. So in one sense, you can consider the genome to be like messenger RNA, and indeed parts of the genome can be immediately transcribed upon or translated upon infection. It's an enormous single-stranded RNA virus, so one of the largest genomes of RNA viruses. And while there are a variety of SARS-like viruses that are related, they do have notable differences. And importantly for my talk today, I do want to remind everyone that the receptor is, so the spike protein on the virus binds to the ACE2 receptor, and the part of the spike protein interacting is the RBD, or receptor binding domain. And so some of the serology we focus on the RBD as opposed to the entire spike. In terms of where this virus came from, there are a lot of different coronaviruses that do cause human and animal disease. We believe this one came from bats. We don't know how it got from bats into humans, but the coronaviruses we're focusing on are the beta coronaviruses, and that's where we have SARS, SARS-2, and MERS, but there are also other alpha and beta coronaviruses that impact humans. OC43 and HKU1 are common human coronaviruses of the same lineage, SARS and MERS, and then we have the alpha coronaviruses 290 and L63. And these four viruses have been causing the common cold for many, many years, and they have been studied, and we have been drawing some of our assumptions for SARS coronavirus 2 from our understanding of the common human coronaviruses, but clearly there are some differences. We do care about gamma and delta coronaviruses, but not for humans. The delta coronaviruses cause a lot of disease in swine, and the gamma coronaviruses are a major problem for poultry producers. And so here in Georgia, we care about the gamma coronaviruses as well. Just quickly about transmission, that's the Ferret model that I am going to be talking about today is a very good model for transmission, although I'm not going to show transmission data. But what we think about is going on with the coronavirus or the SARS-2 is that we have aerosol, respiratory droplet, and potentially fomite transmission. The concern about fecal oral really comes from the original SARS virus that outbroke in the early 2000s, because there was very strong evidence of fecal oral transmission with that virus. And as we've learned over the past year, the details of whether it's a large droplet or small aerosol transmission or fomite certainly can impact the control and spread of this virus. And then also importantly for my talk is the clinical features of COVID-19. And as is widely in the news, there is a large portion of asymptomatic cases. How many there are, we're not certain, but certainly there is an appreciable number of asymptomatic cases. And there's also a large number of mild disease cases. And so these are individuals that may have minor symptoms of fever, fatigue, dry cough, and if they do go to the hospital, they may see ground glass opacities in the lungs and pneumonia. So you do see lower respiratory tract infection. And it's only a small number of the cases where we see severe disease or life-threatening disease. And I do not want to diminish that, but I think that it's important that we acknowledge that the bulk of the cases of coronavirus or infection do cause mild disease. And when we look at that actual host infection, and we'll talk a little bit about that today, so we have the virus that is going to infect the individual, and it can directly infect respiratory epithelial cells in the upper or the lower respiratory tract, depending on the size of the particle that's inhaled or whether it's a fomite. We have the binding to the ACE2 receptor, and we can have either fusion at the plasma membrane or an endocytic event, and then fusion with the endocytic membrane. The genome is released and it can immediately start translating some of the proteins necessary for replication. And then that viral replication complex will enable replication of the full genome, duplication of the genome, the subgenomic RNAs. You get assembly of new proteins, new genomes, and virus release. And so that's where we get our shedding very quickly. At the same time, this is going to trigger the immune responses. So we'll have ingestion of virus, activation of pro-inflammatory cytokine responses. Dendritic cells will be activated. They will take antigen to lymphatic or the lymph nodes. They're going to activate the T cells, the B cells. We're going to get antibody produced, cytotoxic T cells produced, and those are going to control the infection and hopefully clear the infection and elicit immunity. And while the immunity that has been talked about with the vaccines is really focused on B cells and the antibody responses, the T cells are clearly important, and there is growing clinical data that a robust T helper cell and cytotoxic T cell response can be beneficial. So my lab studies host pathogen and pathogen-pathogen interactions, and we've looked at a variety of co-infections, including bacterial and influenza co-infections. As I said, I'm historically an influenza lab, but we've also looked at individual infections with influenza A virus, influenza B virus, respiratory syncytial virus. I did a little bit of SARS-1 research, and now we've been doing SARS coronavirus 2 research. And so early on, we did develop models, and I'll show you some of that data for SARS-CoV-2 infection based upon our work with influenza A and B virus. And what we were interested in is what happens when you have more than one virus infecting an individual, because we don't live in a vacuum. And so those are some of the data that I'm going to share with you today. I always refer to this a paper I really like, and it is not an experimental paper, but it discusses virus-virus interactions, and it puts them into 3 categories of direct interactions where you can have recombination, you can have virus reassortment, so direct activities on the virus, and then you can have helper viruses where you can have co-infections enable replication of viruses that may have defects in their polymerase or other replication machinery. So those are direct interactions. Then we have indirect or environmental interactions, and under this is where we lump innate immunity and the microenvironment. So we could think about the microbiome, we could think about stressors, we could think about the early immune responses that are non-specific. And then lastly, we have, and this paper calls them immunological interactions, which I dislike that term. It is certainly adaptive immunity, but those indirect interactions are also immunological interactions. And so I think more about indirect interactions in terms of innate and adaptive immunity. And there's good data to support the idea that virus-virus interactions may interfere with infections. And so there's epidemiologic studies that have shown individuals that have been infected with one virus have become non-susceptible to rhinovirus infection. Studies from common human coronaviruses in infants have shown that the common human coronavirus infection is negatively associated with other respiratory virus infections. So once they have a coronavirus infection, they're refractory to other respiratory infection for a period of time. Back in 2009, when we had the H1N1 pandemic, rhinovirus infection was reported to delay influenza infection, which in turn delayed RSV infection during that pandemic season. And then in experimental systems, ferrets have been shown that if they're infected with influenza A virus, it can elicit a refractory period where they're no longer susceptible to RSV infection. So that first viral infection prevents a secondary virus infection. And in the case of flu and RSV, those viruses are certainly very unrelated, so we couldn't assume this to be a specific immune response. So the first hypothesis I'd like to present is that the ferret is the best model to study respiratory pathogen interactions. The ferret has been used for getting close to a century now. It was first used in the 1930s for influenza infection, and the first influenza viruses from humans were isolated in ferrets and mice. So they can be readily infected with a variety of respiratory pathogens. I'll share some data there. They can measure pathogen shedding longitudinally because we can repeatedly sample these animals. So we can also measure transmission since they shed virus. And we can readily assess serial infection with a meaningful timeline. I love to work with mice. They're a very powerful tool, particularly for studying mechanism. But with a short lifetime, it's hard to do meaningful studies where you infect with one pathogen or exposed to an insult, allow them to recover, and then look at that secondary infection. And we can measure a lot of things. They're not the powerful molecular tools of mice, but we can measure innate serological and cellular immune responses, as well as look at pathology, some toxicity measures and other things. And that's one of the things I like about being at a veterinary school is we have a robust toolkit of clinical resources for studying disease in animals. And importantly, and the reason that I'm actually here today, I think, is the ferret may also be a good model to assess the impact of PFAS and other environmental exposures. And I'm not going to show you data today. This is something we've just started working on, but there is data showing that certainly with environmental exposures that mink do absorb PFAS and other contaminants, and it can actually alter the health of those animals. And there's experimental data showing that as well. So I'm going to show you two brief vignettes. The first one looking at establishing our ferret coronavirus model and showing that it's useful for other respiratory pathogens. And then secondly, we're going to look at influenza and the potential to interfere with SARS coronavirus infection, as this is certainly very relevant for the flu season that we should theoretically be in the middle of. So the first studies that we set up with the ferrets and coronavirus in the summer of last year were really, others had already shown that the animals could be infected, but we needed to establish the model so we could conduct our vaccine studies and other things. So we asked a really basic question that's important for the work I do, because we look at a lot of different infections in animal models, and it is possible that the form of inoculation, whether it's an aerosol or a liquid, or if we use a different anesthetic, that could alter the ability to inoculate and infect, or potentially the level of inoculation or the level of disease. And so we compared two different anesthetics that we regularly use, an injectable ketamine and an inhaled isoflurane anesthetic, to see if both of these would elicit similar outcomes from infection with coronavirus. And so this is the basic study timeline that we use for all of our studies, where we acclimate animals, we inoculate, we do serial sampling every other day, we collect blood at different time points for peripheral blood mononuclear cells, as well as serologic analysis. We will humanely euthanize and conduct necropsies at different time points to look at pathogenesis. And we can't do enormous group sizes because of just the size of the animals, limited facilities, and the costs, but we do try to have numbers to provide statistically significant differences. To conclude a bunch of data, and this is one reason that I emphasize the disease in humans. We saw no clinical disease when we inoculated these animals with over 5 logs of virus. We saw no temperature changes, we saw no weight loss. We might have had a sneeze or a cough here and there, but nothing remarkable. So the animals did not get clinically ill from being inoculated with SARS coronavirus 2. And we were using the original Washington isolate that was isolated early on during the pandemic. And again, I highlight that if you look at human disease, the majority of cases do not cause severe disease either. And so perhaps if we had infected 100 animals, a small number of those would have seen clinical disease, and an even smaller number may be severe disease. However, we did successfully inoculate the animals. We saw that we've got robust replication when we do nasal washes in the animal, both with the ketamine anesthetics of the injectable and the inhaled. There are really no differences between these, and we saw good shedding for a week. And importantly, early on, we didn't see a large virus, so it's not just residual virus. That's certainly, we wouldn't see that out to 7 days, but we saw an increase and then a clearance over time. So the animals cleared the virus on their own, which is again what we see in most humans. When we look at tissues with the animals that were necropsied on day 4, we found high titers in the nasal wash, in the nasal area, in the trachea, we saw lower levels, but once we down to the lower respiratory tract or in other tissues, we did not detect much virus. With the isoflurane, we did see a little bit in the lung. I don't think this is, again, significantly different. It's more a matter of if we would have done more animals, maybe we would have seen a couple of animals in the ketamine group as well. But again, we're seeing similar to what was seen in humans, distribution of virus not going throughout other tissues. We did PCR on a lot more tissues, and so the PCR data just reflects what we saw with the infectious virus data. We see CTs in the nasal washes. When we looked at fecal swabs, because there were concerns about fecal oral shedding, we saw very little, which others saw as well, although there is a little detectable in the GI tract. And when we did PCR on tissues, we saw a good distribution in different tissues. We even found it in immune tissues like the tonsils and some of the lymph nodes, but not enormous amounts, and it was quite variable. So again, I would argue that this does reflect what we would expect to see in most humans. And after infection, when we look at recovery 14 days later, we did serology, and we found that the animals overall elicited a robust antibody response against both the RBD and the spike protein by ELISA, although we did have one animal that it was quite weak. And again, you see this variability in humans as well, that you have different degrees of seroconversion, with some having high antibody responses and others much lower antibody responses. I do expect this animal 3.1, it would increase over time. This was only at 14 days post-infection, so still quite early in terms of the immune response, but we end these studies quite early since we're in a BSL-3 containment facility. So just quickly, what about other respiratory infections? As I said, the ferret has been used nearly for a century for influenza infections. We've used it extensively looking, and this is just 4 examples. So we've done studies with live attenuated influenza virus vaccines that are temperature sensitive, cold adapted and attenuated, and we can see the viruses replicate very well out to a week. We look at a common human H3N2, we see a slightly different but just similar profile. We see an increase in titer and it's cleared by a week, again similar to what you see in humans. A Switzerland virus from H3N2 in 2013, we see high titers very early on and then it's cleared by a week. And then lastly is Singapore virus, and again it's another H3N2, and I emphasize this one because this is the virus we're going to be using for our co-infection studies, and we see that with inoculation we get a robust infection and titer that again clears in about a week. In all of these cases, the H3N2s don't cause significant clinical disease in ferrets. It replicates very well. We get good seroconversion, but it doesn't cause severe disease. Looking just quickly at other viruses, so RSV, as I mentioned, this also replicates very well in ferrets, and so it provides a very useful model for looking at potentially flu and RSV infection, as has been reported in the literature. And then On the left or on the right here, common human coronaviruses, so OC43, we just very recently demonstrated that you could inoculate ferrets with OC43, and while there's not very high titers, we do get consistent infection, increases in titers, and then clearance over a week, which is actually quite powerful because, to my knowledge, there are not any good animal models for the common human coronaviruses. And so this is going to be useful for our future studies to look at how repeated coronavirus infection may affect outcomes with SARS-CoV-2. But this is very early days for that. And as I mentioned before, we've done co-infections as well, and this is just an example where we've infected animals with strep pneumo and allowed them to establish a baseline infection with strep pneumo over several weeks. And then we infected those animals with the influenza, in this case an H1N1. And what's notable is, so here's the plaque forming units of influenza coming up and clearing over time. If you look at the colony forming units for the strep pneumo, it was very, very low, about 100 CFU available to these animals when we first inoculate them with flu. And once they're infected with flu, we see this expansion of the bacterial infection, which is another very powerful model for looking at co-infections, where respiratory infection may cause expansion or an opportunistic pathogenic infection from bacteria. So just to quickly sum up this part, I want to mention that SARS-CoV-2 is replicates well in the ferret. I think we have a very good model, albeit we don't have disease, but I think it's representative of a lot of what we see in humans. We also show that we have other very useful infection models, and for these reasons, I think that the ferret is probably the best model to look at virus-virus interactions, because we can infect them without having to adapt viruses, which you have to do with mice in general. So we can naturally infect animals and look at those interactions longitudinally with serial sampling and get meaningful data on virus-virus interactions. I see you coming on, Megan. So the last part is short. So what will happen this year with the flu and coronavirus? I'll quickly show you. This is what happened 2 years ago with flu. We have a very robust flu season. Last year, we had a very robust flu season until we had the shutdown, and it went away. And if we look at this year, here's our flu season to date. Less than 1% positive tests, less than 150 positive specimens in the United States, whereas we had tens of thousands in the past 2 years. So we wanted to test the question, what happens if animals are exposed to flu A and then to coronavirus? And so we had groups of animals at different times inoculated with flu, and then we'd inoculate them with coronavirus. We really didn't see a lot of clinical changes in terms of weight loss. We didn't see any differences in temperature. We saw some weight loss in some groups, but that appeared to be unrelated mostly to the coronavirus infection. And so our inflammatory response analysis may tell us more about this, but at this point I'm saying we didn't see any changes in clinical disease. Looking at virus shedding, though, we see a very interesting story. So here on the right, here's our naive animals to flu. We infect them with coronavirus. We see good replication, although variable over time, and they clear it by about day 7. If we infect animals with flu here on the left, here's flu titers, 11 days, 9 days, and 7 days before SARS. We inoculate them with SARS, and notably, only one animal had on one day a very low detectable infectious titer. So this flu infection, 11 days prior to SARS infection, prevented flu infection almost completely. And at this point, I'll note that we, for almost a week, had no detectable flu virus. If we move a bit closer to 7 days prior to, so here's our flu infection, we have complete inhibition of SARS coronavirus infection. If we go to 3 days before, we see a very similar pattern and spike in flu, although interestingly, it appears to be cleared earlier and absolutely no SARS. If we co-infect at exactly the same time, we actually see that both viruses replicate about to the same level, maybe a little bit lower, so it is not an absolute interference where the viruses can't coexist in the environment. And if we flip it and we look at SARS first, so here's our SARS infection, we inoculate with flu 4 days later, and we see absolutely no flu. So the presence of the SARS infection prevents flu, the presence of the flu infection prevents SARS, and notably, even after the flu is cleared, at least for 6 days, the animals are still pretty much refractory to infection with SARS coronavirus. We do see seroconversion to match those data. So when the animals did not have replicating SARS, we don't detect SARS titers as shown by the area under the curve analysis. The one odd one is we saw no detectable serum antibody responses during the co-infection, although we did see virus, so we're confirming this with other assays. And we see the same thing with flu. When they're infected early, we get good flu titers by hemagglutination inhibition assays. And then we just have one odd one that although we did not detect infectious in replicating flu on day +4, we still do get some seroconversion, although lower. This could be because the antigen in the inoculum, and we're testing that now by using an activated virus. But we do see generally serological patterns that reflect what we saw with infectivity. So we have continuing studies. These data are really coming off on a daily basis. We are looking at the inflammatory response in those nasal washes. We need to confirm the virus load by PCR, and we did confirm some of the serological responses. But I can conclude at this point that we do see interference for 11 days. There's a lot of possible mechanisms. Maybe it is remodeling the cellular architecture there, although I think that's unlikely. It could be an alteration in the microbiome. That's certainly a strong possibility. It could be loss of susceptible cells from innate responses. I think that's probably the most likely case, in that we're seeing a continuing inflammatory response due to the innate immune response, and that the pro-inflammatory cells, that pro-inflammatory response driven by the innate cells, including NK cells and neutrophils, may be either rendering cells refractory or directly inactivating coronavirus just through the inflammatory environment. And of course, there could be other mechanisms that are exploring as well. So thank you for your time. I'll just put my acknowledgements up again, and I welcome questions from the group, and I'm very happy to be here. Thank you.
Thank you very much, Dr. Tompkins, for a very interesting presentation. I'm going to encourage the audience to reach out to Dr. Tompkins during the spatial chat networking period. I'll sneak in one quick question that addresses multiple questions that we've gotten so far. And you've demonstrated how this model is successful at looking at co-infections. How do you see the impact of environmental exposures, or how could you tie in the exposome into this model? And some of the questions specifically relate to things like PFAS and to air pollution.
So for PFAS in particular, the mink has been studied looking at environmental exposures to PFAS molecules, and they've shown that you can have alterations in weight gain, you can have liver toxicity, and potentially alterations in immune responses. And so that's where we plan to focus in looking at how does exposure to PFAS impact coronavirus infection just as a mono-infection, but also how does that alter serial infection with different viruses, particularly looking at innate immune responses and adaptive immune responses. And those are things that we can measure quite effectively in the ferret, looking at changes in the magnitude of the antibody response, the magnitude of the T cell response, and the magnitude of the inflammatory response, or possibly the skewing of those responses. And so again, I think the ferret provides a meaningful model because we can have extended periods of exposure, we can measure that toxicity very efficiently, we can measure the PFAS levels very efficiently, and now look at the questions of how that influences outcomes from infection and immune responses to infection. And importantly, we can shift this over to vaccination as well.